Hi, my name is Jung. Thanks for having me today. So today I'm gonna introduce you a novel rising non-invasive brain stimulation technique called transcranial focused ultrasound stimulation, and how it can used in the cognitive domain to modulate the targeted cognitive processing. I am particularly interested in the semantic memory, so that's my target cognition. So, transcranial focused ultrasound stimulation is relatively new and innovative and also very, very high, well, I think still very highly promising non-invasive technique. So the different from existing TMS or TACS or the TS, it generates low acoustic energy, and then compared to the existing the techniques, it can directly stimulate the part of the brain because acoustic energy can penetrate into the our brain, and also it has the more high spatial resolution compared to TMS. So the TMS is like a centimeter, like 1 centimeter each, but now as you can see in this figure, it goes to typically 5 to 8 millimeter with the novel transducers. So now it's very, very, very tiny. So all these strengths and advantages make this technique is very promising. And then that is the ultrasound equipment. Also CPU has it. So the main body is a small box, and the bottom one is this is a transducer. It's generating the acoustic energies. So within the transducer, there are several layers of the acoustic generators. So by modulating the angulation of those generators, we can generate focused ultrasound energy with different depths. So currently the technique is on highly lots of the investigation going on. For in animal work, it showed very promising result by modulating the opening of blood-brain barriers, as well as in human work, it's generated the neuromodulatory effect like TMS and then TS. So what is the underlying mechanisms so far? Still under research, but these are the agreed, the underlying the mechanistic effect of ultrasound. So ultrasound stimulation excites neurons through the actual physical effect. So when it hits the membrane, it's gonna rise temperature, very subtle, not fatal. And also in animal model, it shows the bubble formation and cavitations, but non-human reported it. And then last bit is it actually vibrating the membranes. So these physical effects, they are co-occurring. So all together, I'm not gonna go through the chemical changes. So eventually, it's gonna modulate the openings of the certain ion channels, which generating the neuroplasticity effect. So this is the latest review paper summarizing all published work. That was the last February. So compared to last year review paper, now the number of studies, I think, 4 times more. So the top one is healthy human studies. So it started with the typical central motor cortex, but now in the medial view, you can see there are studies sonacating amygdala, entorhinal cortex, thalamus, hippocampus, basal ganglia, so the key deep neural structures. And now there are lots of the clinical studies published. Also, there are ongoing clinical trials for epilepsy, Parkinson, dementia, typical psychiatric disorders, ADHD, depressions. You can just name it. You can find the preliminary result. And then now you see they are stimulating the nucleus accumbens and the ventral striatum, which is the core neural circuits associated with diverse clinical disease. So when we sonacate the human brain, we need to think about skulls. So from this example, you can see everyone's face. So the skull's shape and then their shape and densities are various. This is the core thing to deliver the ultrasound stimulations. Well, it's similar to the TMS field modeling. Of course, someone had a thicker skull. The reach the intensity of the ultrasound is gonna be lower compared to the thin skull person. And also the skull structure is not homogeneous. So when you see the magnifying the skull has several layers. So there are some airs within the skull structures. And when we deliver the ultrasound, air is one of the big obstacles because it reflected almost 90% of the ultrasound energies. So therefore, when we actually deliver the ultrasound stimulation with the intended protocols, we need to perform the simulations similar to TMS field modeling, and then we have the commercialized software called the K-plan simulations. But our group also suggested free open source to get the entry point of the transducer, considering the shortest distance from your target area, and it's gonna try to find the thinnest skull as well as flat as possible. So they'll provide 2-3 entry point of the transducers. So in the actual experiment, you can test and try different entry point of the transducers. So currently published, luckily, and then it is open source. With the T1 image, it's gonna take 10 to 15 minutes to get processed. It's gonna generate you to 2-3 entry point of the transducer, and then kind of give you the shortest distance. So now I'm gonna talk about shortly about the semantic memory. So from neuropsychology and neuroimaging studies, we agree that the anterior temporal lobe is a semantic transmodal hub. So they're located the ventral anterior part of the anterior temporal lobe. And using MRI spectroscopy and then the task fMRI, so those colored blob within the anterior temporal lobe, so the GABA signal within that box cells are positively correlated with the semantic task performance from my 2 different studies. And more importantly, that hotspot is also nicely overlapping with the ECOG study from Chen et al. So that's our target area. So obviously it is located in the slightly deeper part of the anterior temporal lobe. So the aim of this project is I'd like to enhance semantic memory in healthy populations. So we chose theta-burst focused ultrasound protocol by Jung et al. So this is mimicking theta-burst TMS protocol, obviously, with a more higher frequency of the ultrasound. So it's 8 seconds train of the 20 milliseconds burst of the ultrasound repeated every 200 milliseconds. So in motor system, it significantly increased motor evoked potentials compared to the just repeated pulsed stimulation and sham stimulation. And more importantly, in the motor simple execution task, after the theta burst focused ultrasound stimulation, participants were actually faster. And this protocol has been used in other cognitive domain. So when I had this protocol over my right IFC, my performance in stop signal task was, I think, roughly 20% improved. So it's very, very effective. So far, the effect is replicated in different tasks in humans. So I chose to use this protocol. So this is the overall study design. So to have the ultrasound target, we must acquire the T1 image first. Then participant received either the left anterior ultrasound stimulation and ventricle as a control ultrasound stimulations. So in the day of testing, participant performed the semantic association task, which is a Cameron Cocktails test, and we have a matched control task, which is a pattern matching task. Then we sonacated ventral anterior temporal lobe, which is one participant example here, and or the ventricle stimulation which is also located in the left hemisphere here. Then they enter the MRI scan, so take kind of 5 minutes. I acquire the structure image, MRI spectroscopy, to measure and quantify neurochemicals at the left anterior temporal lobe as target. Also, occipital cortex was a control regions. Then participants also performed the task fMRI, doing exactly same task inside of a scanner. Then it's long, it's a very prolonged session. When they got out of the scanner, I kindly asked them to repeat, please do this task one more time. So it's gonna take roughly 2.5 and 3 hours, depending on participant performance. So I just got this figure yesterday. So this is the average, the acoustic field modeling across all the participants generated from the K-plan. So in the left anterior temporal, as you can see, the hotspot is here, which successfully stimulated the target area, and even for the ventricles, hopefully you can see the overlapping. Yeah, we are stimulating the CSF non-brain area. There is no brain region. So in terms of the ventricle stimulations, we verified ventricle stimulation does not generating the neuromodulatory effect. If you're interested in, this is a QR code for the preprint. Hopefully can publish in this month. So overall, the reached acoustic intensity is around 60 and 70% of the intended protocol, so that we are pleased about the simulation result. So I'm gonna show you the quick demonstration of the actual ATL stimulation from my friend as a guinea pig, the first pilot.
1, go.
So when my colleague pressed the start button, now sonocation is starting. Compared to TMS, no sensory effect, very quiet, nothing's happening.
Is it okay or pressing fine? Okay.
And participants, they don't feel anything at all typically. So again, another advantage of focused ultrasound is it's a minimal side effect compared to TMS. So it's very, very comfortable. So both of experimenter and then the participant, they do not feel anything at all during the stimulation. And of course, you see lots of the ultrasound gel applied at the target area. So it goes on until the 80 seconds of stimulation.
Is micro movements a problem?
So is it actually effective? So let's see the behavior result first. So the green, not green, the cyan color is the ATLs on occasions, and then black is ventricle stimulation, and then the triangles and circles are the individual participant performance. So after the ATL stimulation, compared to the pre-session, participant improved the task performance, roughly around 5% accuracy increase. That was also significant compared to the control stimulation. And in control task, which is a pattern matching task, there is no behavior effect. So I was very excited and pleased about the effect compared to TMS. I've done exactly the same study with RTMS, TVS, TDCS. This is the biggest behavior effect I ever get through last 10 years of experience. So very effective. And remember, this is a healthy young participant. So let's look at the neurochemistry. So ATL stimulation significantly decreased the local GABA concentration, as well as increased the local glutamate concentration at the target area. And apart from these neurotransmitters, we also observed that there is increase of the N-acetylaspartate, which is the neuronal marker for aging and then dementia. So if we get older, have a dementic brain, NAA is decreased, and then also we found a significant increase of choline. So ultrasound, its mechanical effect resulted in the the full changes in neurotransmitters as well as neurochemicals at the target area. And this effect is transient, so that we comparing the same participant 5 days apart. Then, more surprisingly, when I analyzed the structure image using VVM, ATLs on occasion significantly increase the regional gray matter at the targeted brain area. So again, this is a transient, so it's 5 days apart, the same participant. So I was very surprised about this gray matter density increase, but I have some explanation, luckily. Okay. Now, when we look at fMRI, based on the local neurochemical changes and then brain morphometric changes, of course, fMRI board signal was significantly modulated by the ATL stimulation, not only at the target area, but also all connected semantic network, bilateral hemispheres. Again, this is the biggest effect I ever had compared to the TMS studies. So to understand about the more network perspective, I performed dynamic causal modeling, which provided the directional connectivity changes driven from the experimental conditions. So I set the 6 regions of ROI as a semantic network, and then this is when there is no perturbation in the semantic system, everything is positively connected, which means all the semantic regions are facilitatory to each other. However, after the ATL stimulation, now all the connections were significantly increased. Everything was significant statistically. And just to focus on the ATL stimulations, so I took average of the ATL stimulation to the ATL target region from other semantic node or from ATL to the rest of semantic systems. So roughly, we observed 8-fold increase in semantic network intrinsic connectivity following ATLs on occasions compared to the control stimulation. So again, the biggest neural effect I ever observed in this field. So hopefully, I can convince you this technique is effective and safe with minimal side effect. So focused ultrasound can enhance semantic memory by showing task and region-specific effect. Specifically, it enhances semantic memory performance by modulating neurotransmitters and neurochemicals at the local level. Also, there is an increase of the gray matter density and neurodynamics of the entire targeted semantic network. So therefore, I hope I can convince you this technique is emerging as a very promising therapeutic tool to enhance memory in patients with dementia with minimal side effect. And then, well, the last bit is a very obvious one. So this type of approach provides a convergent understanding of the neurochemical mechanisms of human cognition and its neuroplasticity. So I'd like to thank all of my collaborators and thank you for your attention.